Hello, everyone. Welcome to another GRiP-ing seminar. Before I let Richard introduce our speaker today, just a couple of announcements. There's no talks for the next couple of weeks, Saturdays. And we'll be meeting again on March 13th for another GRiP-ing seminar from Joanna Santos from I2BC Perry. All right.
Thank you. Okay. It's great to have Kirsty McWilliam here. Kirsty is a graduate from former Parasitology undergraduate degree, graduated in 2015. I first met Kirsty at the Millport field course.
Yes.
It brought to my attention that she liked cycling, which is actually I introduced myself to her. Anyway, she did the MSci, I think, the year after that, and went to MSD as it was then.
I did, yeah.
Hosted by Paul Seltzer.
Yes.
Yeah, and then finished her degree and went to do a PhD with Keith Matthews in Edinburgh, and then to do a postdoc with Nikolai Siegel, and then back to Keith's lab, where she is now. So,
yeah.
to you, Kirsty.
Oh, great. Thank you very much. Thank you everyone for coming today. I know I'm not nearly as qualified as the people normally are that come and present these seminars, but I will do my best. And it's cool being in this room actually, because I'm pretty sure this was the room that I did my presentation for my undergrad project as well. So funny being back here. Great. Okay, so what I'm going to be talking to you today about then is the project that I've been doing for the last 4 or so years in Keith's lab, where we've used a genome-wide fitness profiling approach to identify novel regulators of Trypanosoma brucei life cycle development. And I am going to start a timer. Probably should have done that. There we go. Okay. So one of the trypanosomes that we're interested in Keith's lab is Trypanosoma brucei. And as everyone in this room will know, T. brucei is a single-celled eukaryotic parasite that lives, there we go, extracellularly within their mammalian hosts. And they are transmitted by their tsetse fly vector. Now, whilst human cases of African trypanosomiasis in sub-Saharan Africa remain low, animal cases certainly don't. And animals that are infected with African trypanosomiasis present with weight loss, anemia, and as such have greatly reduced productivity. And therefore the animal disease has considerable economic impact in the region. Now, when the trypanosomes are within their mammalian hosts, they undergo a developmental transition. And here, slender form parasites, which are proliferative and very important for establishing infection in the mammalian hosts, slender form parasites differentiate to these stumpy forms. Now, stumpy forms are cell cycle arrested in G1, and this is important for those early phases of parasitemia so that the host doesn't become overwhelmed by the parasites. These stumpy forms are also pre-adapted for transmission into that tsetse fly vector. Now, this differentiation from slender to stumpy forms is density dependent and is very much reminiscent of bacterial quorum sensing. So what we knew for a long time was that as the number of slender forms increased, so too did the concentration of something that we knew as stumpy induction factor, or SIF. So the number of parasites increases, the concentration of SIF increases, until it reached some kind of like tipping point, and then these slenders differentiated to stumpies. So this was kind of our understanding for many years. However, a lot of work, I've highlighted some of the papers down here, it means that we now kind of understand this quorum sensing differentiation process in a lot more detail. So what we know now is that parasites release peptidases into their extracellular environment, and these peptidases generate oligopeptides. These oligopeptides are transported into the trypanosomes via the cell surface transporter GPR89, And then this triggers a signal transduction pathway that then results in stumpy formation. Now, many of the components of this signal transduction pathway were identified in this Mone McGregor et al. Study here. And what they did was they used a genome-wide RNAi approach. So this was using the GRiP-ing technology that was available at the time. And they grew the cells transfected with the RNAi library, with the RNAi induced, and then they exposed the trypanosomes to a differentiation trigger. And then if a gene that was part of this signaling pathway was knocked down, it would mean that the parasites wouldn't respond to this differentiation trigger. They would outgrow within the population, and then you could sequence these and find out which gene had been knocked down. So that's how many of these had been identified. And as successful as this study was that identified these components, there were limitations. So first off, this differentiation trigger that I was talking to you about, what they had used was a cell-permeable cyclic AMP analog. Now, because the cyclic AMP analog is cell-permeable, this meant it completely bypasses GPR89 and all those very early signaling components. So this means that they weren't able to identify anything that was happening in those kind of earliest events. The screen also made use of a monomorphic cell line. It was all the GRiP-ing RNAi library was available then. But so monomorphic parasites have a reduced capacity to differentiate from slender to stumpy forms. And this is different to the pleomorphic cell lines that we like to use in Keith's lab, which are able to undergo the slender to stumpy transition. So the aim of my study then was to try and identify these molecules that were involved in these earliest quorum sensing signaling events using a physiologically relevant screen and a differentiation competent cell line. So to do this, we turned to Kat Gadelha and her team in Nottingham, who alongside Bill Wickstead have developed this direct RNAi fragment sequencing approach, or GRiP-seq. And what GRiP-seq is a powerful genome-wide RNAi approach, but one that rather than just giving a kind of binary, these cells dropped out of the screen or these didn't, instead gives us a quantitative measure of gene fitness across the entire genome. And importantly for us, the library is also available in differentiation competent pleomorphic cells. Excuse me a second. Okay. Cool. So for this new screen then, what the team in Nottingham did was grow the parasites transfected with that GRiP-seq library for 72 hours with the RNAi induced. And this was either done where there was no selection made on the parasite, so this would give us just a general gain or loss of fitness profile, or where the cells were grown in the presence of BHI. This is brain heart infusion broth. And BHI is important because it contains the source of external oligopeptides. So it contains that physiologically relevant differentiation signal from the oligopeptides. And here we were asking then, so knockdown of which genes will decrease the response to this quorum sensing signal? So here's an example of the results from one of the two replicate screens that we did. So each of these little dots here represents a different gene. And on this axis here, what we're looking at is decreasing fitness upon gene knockdown. And then that's in the conditions where there were no selection. What we're looking at on this axis here is increasing levels of insensitivity to BHI upon knockdown. So basically, if you knock one of these genes down here, the cells are less responsive to BHI. So what we were interested in were the genes that had appeared in this part of the plot here, where we saw that knockdown conferred the strongest gain of fitness phenotype that was specific to those plus BHI conditions. And if we mapped previously identified quorum sensing components onto my results here, so they're in blue here, whilst we did see that many were kind of concentrated in this blob in the middle, we did see that these three kind of came out in exactly that region of the plot that we were looking at. And these three, they've all been validated, and we know that they're important for slender to stumpy differentiation for the regulation of this. So this gave us a bit of promise that, right, our screens maybe worked all right, we're interpreting the results. Okay. And we've pulled out a number of molecules from the screen that we're interested in, but I'm going to be talking to you about these two kinases here today that have been the kind of best hits that have come out from the screen. So we've got this AKT-like kinase up here, and then this AGC family kinase here, which I'm just going to call AGCK for the rest of this talk. So the first thing to do is actually to validate, okay, does knockdown of either AKT-like or AGCK actually make the cells less responsive to oligopeptides? So the first thing I did then was to generate independent RNAi lines targeting either AKT-like or AGCK, and then follow their growth in vitro in the presence of oligopeptides. And again, we did this by growing them in medium supplemented with BHI. So what we would expect then when the RNAi is off and the signaling pathway is complete, the slender cells should respond to the oligopeptides in this BHI, arrest their growth, and then differentiate to stumpy forms. Whereas when the RNAi is induced, we'd expect that now the signaling pathway isn't complete, and then these cells will continue proliferating. They won't respond to those oligopeptides. So what we saw is this. So if we look at this bold line at the bottom here, these are the cells, the RNAi lines, when they're uninduced in the presence of BHI. You can see them arresting their growth here. Whereas when we induce the RNAi against either of the kinases, you could see that the cells became less responsive to that BHI. So that kind of validated that, right, we're interpreting our screen correctly here. But as you can see from, so these are QR2-PCR plots here, particularly for AGCK down here, my levels of knockdown weren't fantastic. So we thought, right, we'll generate some knockout cell lines and move forward with these. So that's what we did. I made double knockout cell lines for either AKT-like or AGCK, and then we repeated those assays again, following their growth in vitro, grown with the BHI. And again, what we'd expect from our parental cell lines, that they should respond to the BHI, arrest their growth, whereas for either of the double knockout cell lines, that they should become less responsive to that BHI. And if we look here, so we've got this dotted line down here. So this is the parental line. You can see arresting their growth. And when we knocked out either of the kinase, so AGCK or the AKT-like kinase, again, you can see the cells are responding less to the BHI. So that was great. But if you were sat there thinking, well, there's a lot more to mushed up brains and hearts, essentially, than just oligopeptides, then you would be absolutely right. So to really hone in on that oligopeptide signaling, we teamed up with Professor Mark Vendrell and his postdoc, Lorena Tapia, in Edinburgh. And what we did was to design and synthesize, I say they did all the synthesis, synthesized targeted dipeptide sublibraries. And then we screened in these dipeptide libraries to find dipeptides that more efficiently induced slender to stumpy differentiation. And as the title of my slide completely gives away, the best dipeptide that we found for inducing slender to stumpy differentiation was this one here. So NF and asparagine followed by phenylalanine. Now, if we grow pleomorphic cells with this NF dipeptide, so if we just look at their growth here in vitro again, you can see the pleomorphic cells arrest their growth in response to the dipeptide, and then they adopt this beautiful stumpy morphology. They really are gorgeous looking cells. And they are expressing this stumpy-specific marker protein PAD1 on their surface. So they really are kind of differentiating to stumpy forms here, rather than this just being some kind of generalized toxicity. If we grow monomorphic parasites, so remember those parasites that can't efficiently undergo that slender to stumpy differentiation process, you can see that the monomorphic parasites, they're not responding at all, don't arrest their growth. And then if we look at their PAD1 surface expression, there's none. Cool. So now that we know that we're definitely looking at dipeptide that induces slender to stumpy differentiation, we now took either our AKT-like or our AGCK double knockout cell lines and grew them with the dipeptide. And what we saw was that when we got rid of either of the kinases, that now the cells were responding less to this dipeptide. And if we look at PAD1 surface expression by F, you can IFA, sorry. You can see that either the knockout cell lines, they weren't expressing PAD at all, or there was greatly reduced PAD1 surface expression. So yeah, we're really linking these two kinases, AKT-like and AGCK, to that oligopeptide signaling response. So that's all great, but at the moment we're still in vitro. So to really kind of ask whether AKT-like and AGCK are involved in slender to stumpy differentiation, we moved in vivo. And we infected mice with either the wild type or the double knockout cell lines, and then followed the progression of the infection. If we look at the morphology on day 7 of the infection, you can see that the parental cell line, again, is differentiated to these stumpy cells here, whereas for either the AKT-like or the AGCK double knockout cells, you can see they're enriched actually for cells that have maintained this long, slender morphology. And I've told you already that stumpy forms are cell cycle arrested and that they expressed PAD1 on their surface. So again, we did IFA and looked for that across the infection. And in yellow here, well, here in general, sorry, we're looking at actively replicating cells. So in yellow, the parental cell line, you can see across the course of the infection that the proportion of cells that are actively replicating is decreasing during the course of the infection. Whereas for either of the knockout cell lines, so AKT-like in pink or AGCK in blue, you can see that staying relatively consistent across the course of infection until that final day where you've got a slight drop. And again, if we look at PAD1 surface expression, again in the yellow, you can see the parental population, they're almost all expressing PAD1 by the end of the infection, where we've got a greatly delayed response in those AKT-like and AGCK double knockout cell lines. So together then, yeah, we can say AKT-like and AGCK, the knockout of either of these delays slender to stumpy differentiation in vivo. And yep, we're using the double knockouts. Of course, we now need to do the add back experiments just to validate that. So for this, I added back either a single copy or 2 copies of either of the kinases back into the endogenous locus and asked if this restored differentiation competency in our cells. And what we're looking at here is the percentage of cells that are expressing PAD1 after growth in BHI in vitro. So in the yellow, we've got the parental, and you can see a big reduction in cells expressing PAD1 on their surface in the double knockouts. But then when you can see when we add either 1 or 2 copies of the gene back into that endogenous locus, they would restore that PAD1 expression back to the parental levels. So cool. So we found 2 kinases. We're sure that they're definitely they're involved in slender to stumpy differentiation. So the next step then was to ask, well, what are these 2 kinases and what are they actually getting up to in the cells? So for this, we've really focused in on AKT-like, just because we can actually kind of see some domains and stuff. AGCK is a little bit more elusive. We can talk about that at the end if anyone wants. But yeah, so AKT-like, it's a serine threonine kinase. It's got this plexion homology domain and its N-terminus, so these are important for binding membrane lipids. N-terminal pH domain, essential kinase core, and then the C-terminal regulatory domain. And now AKT in mammalian systems is an exceptionally well characterized molecule. And we know it's a real kind of central node for a huge number of signaling networks. So we could really learn a lot from these different studies. So what we know from these systems is that AKT exists in its inactive conformation in the cytosol. So its pH domain is kind of folded over on top of it. But following production of PIP3 at the membrane, you then get relocalization of the kinase. The pH domain binds this PIP3, and again, it undergoes this conformational change where now the kinase kind of opens up, and these two phosphosites are now exposed. And these two phosphosites are important for the full activation and activity of the kinase. So for our functional study of T. brucei, AKT-like, we did a mutagenic study whereby I took that AKT-like double knockout cell line, and then I added back either a single wild-type copy of the gene, just like I'd done before, or I added back a single mutated copy of the gene. And then we asked, how do these mutations affect the cell's response to oligopeptides? So do these mutations make an inactive kinase or not? So what we're looking at here is just our, so again, we're doing this in vitro with our BHI. So yeah, what we're looking at here is plus BHI growth relative to minus BHI. And again, you can see, so our wild type cells respond, less of a response in that double knockout cell lines, and then the cells are beginning to respond again in either with 1 or 2 copies of the wild type AKT-like is added back. So the first mutations that we made were ones which we predicted would be important for the kinases. Ooh, catalytic activity. So that was a very exciting one to build up to. Yes, so we mutated in the ATP binding site here or within the catalytic loop. And then when we look at either of these, you can see that we haven't restored that response to BHI. So we've got an inactive kinase there. We next mutated these two phosphosites here. So we've got T290 and T450. And we believe these are the equivalent of those two phosphosites that I showed you in the mammalian systems, and they're important for the kinase's full activation and activity. And if you can see, I can't see, and I'm all the way up here. There we go. So we made a phosphodiesterase mutant for this T290 site. And you can see for that phosphodiesterase mutant, so a non-phosphorylable form of the kinase, that this didn't restore that BHI sensitivity, not so much for the T450 site. So yeah, we really think this phosphosite here is particularly important for the activity of the kinase. And indeed, when we made the phospho-mimic versions for either of those two sites, you could see again that the cells are responding to that BHI. To investigate the pH domain, two different approaches. So the first one was just to lob the entire pH domain off, see what happens there. So when we took the entire pH domain off, what we actually see is that the cells are still responding to BHI. So I think is happening is here. So if you remember, I was saying this pH domain is important for hiding those two phosphosites that are important for the kinase's activity. So by taking off that pH domain, we're kind of leaving those two phosphosites available, or yeah, they're exposed, and this the kinase can still be phosphorylated here and activated. So to try something a little bit more sophisticated than just taking off the entire pH domain, I mutated this residue here. And this residue, certainly in T. brucei's AKT-like, it's been modeled to interact with those membrane lipids and be important for the interaction there. So I mutated that to try to disrupt this interaction. And indeed, what we saw was with that mutant there, that we didn't see a restoration of that response to BHI. So together then, yeah, we think these AKT-like catalytic residues is lipid binding ability, and particularly these two T290 phosphosites are all important contributors towards a cell's oligopeptide response. Cool. So just to recap that before we move on. Cool. So we found these two kinases, AKT-like and AGCK, by DRIF-seq. We know that when we knock them down or we knock them out, the cells are less responsive to external oligopeptides in vitro. And we also know that if we knock either of them out, that we delay stumpy formation in vivo. And as I've just explained, we think that AKT likes catalytic residues, lipid binding ability, and this phosphosite here are all important towards the cell's response to oligopeptides. But let's go right back to that original aim that I told you about. Remember, where did we go? There we go. We're looking for molecules that are positioned here on the signaling pathway. So how could we place AKT-like and AGCK on this quorum sensing signal transduction pathway? Well, again, let's go back to the start and think about that cyclic AMP analog. So remember, I told you that the cell-permeable cyclic AMP analog, it triggers the quorum sensing response. However, because it's cell-permeable, it bypasses all of the GPR89 and all of the early components. We also know that if we RNAi, if we knock down or we knock out all of these known components that are further downstream here, we make the cells less responsive to that cyclic AMP analog. So, if AKT-like and AGCK are upstream of where cyclic AMP triggers the quorum sensing response, we would expect that if we grew either of our double knockout cell lines with a cyclic AMP, all this downstream signaling pathway is still intact, so the cells should arrest when we treat them with the cyclic AMP analog. Whereas if AKT-like and AGCK are downstream of where it triggers quorum sensing, then we would expect that our double knockout cell lines would actually continue proliferating when we treat them with a cyclic AMP analog, because now the pathway's been blocked somewhere. So obviously, we're a little bit biased. We want to see them arrest when we treat them with the cyclic AMP. It's a little bit early and coming on to the good result. Okay, so here's what happens with the parental cell lines when we treat them with cyclic AMP. So in the dotted line here, you can see that they are arresting their growth here. And as a control, we used a DIRC knockout cell line. So DIRC is positioned here. So as part of that downstream response. And if you knock out DIRC and then treat the cells with a cyclic AMP, you can see that they're not responding. They continue proliferating. So what about AKT-like or AGCK? Well, if we treat them with cyclic AMP, so either double knockout cell lines, you can see that just like the parental cell lines, that they are arresting their growth, meaning that they are upstream and exactly in that region where we were looking at. So that was really cool. For once, one of my ideas for a cool experiment actually paid off. It worked. So we know AKT-like and AGCK, they're now upstream of all these components, but then that, of course, just raises the next question, right? So how are these linking up with the rest of the signal transduction pathway? What's happening here? So again, for this, we just focused in on AKT-like, again, because we knew more about the molecule, and we could kind of use what was known from other model systems to maybe try and guide us in our search here. So what we wanted to know then was, what is this kinase AKT-like phosphorylating during differentiation from slender to stumpy forms? So to do this, we went down the route of mass spec-based quantitative phosphoproteomics, and we did this using in vivo derived cells, just to make life complicated for ourselves. So what we did then was we infected mice with either wild type cells or AKT-like double knockouts. And we took samples either from the ascending phase of parasitemia, so early on when we wouldn't expect any differentiation to be occurring, or we took cells from the peak phase of parasitemia, this later phase, where we would now expect commitment to be occurring. And then we could ask here, okay, so what is getting phosphorylated in these wild type samples that isn't when we've knocked out AKT-like? So that's exactly what we did. Here I'm just showing you a PCA plot that was generated from the phospholipid data. And what you can see is that our three replicates they're grouping together reasonably well, considering these all came from separate mice. And yeah, you can also see though that the separate conditions are forming their own groups. So you can maybe say we've got a bit of a double knockout wild type split here. certainly that we've got our earlier ascending samples here, and then as things differentiate, we're moving over to the right. Certainly won't interpret any more than that from that. And if we look at the abundance of our phospholipeptides in each of these different conditions, we can start to see patterns forming. So for instance, we've got ones like these here, where we've got this kind of low signal in the earlier ascending groups, whereas at the peak later samples, you can see that now we've got more of these phospholipeptides, but that's in both the wild type and the double knockouts. We can see the opposite here, for instance, where they're higher in ascending, lower abundance in peak, again, for both the wild type and the knockout. But then you can see groups such as these, where we always seem to have more of these phospholipeptides when we've deleted the kinase. We could be looking at some kind of indirect effects here. But then ones such as these down at the bottom, which you can hopefully see, which is where we've got less of these phospholipeptides whenever we get rid of AKT-like. So these are the kind of ones that we're interested in. These are our potential substrates, perhaps, of AKT-like. So here I'm just showing you the differential analysis between our AKT-like double knockouts versus the wild types. On the left, either in the ascending or on the right in the peak phase of parasitemia. And these in blue are where we've got significantly less of these phospholipeptides when we've got rid of AKT-like. And you can see that in the earlier time points, in the ascending phase of parasitemia, we've really not got that much difference going on. Whereas at this peak phase of parasitemia now, we've got a lot more differences going on here. So we've got over 100 phospholipeptides that we see significantly less of when we get rid of the kinase. But look, we've got cells that have come from an in vivo infection, right? There's going to be a lot of things that are going on in this 104. We're going to have a combination of indirect and direct changes. So how can we try focus in on those AKT-like substrates? Well, again, this is where we could kind of look into the literature, see what's been done in other model systems with AKT. And what these have all reported is that there is this canonical AKT substrate motif here. So AKT recognizes and phosphorylates this motif here. So this meant what we could do then was look within our data and see, have we got any differentially phosphorylated peptides in this 104 that have got this canonical AKT substrate motif. So that's what we did, and we found 3. So I've got just the peptide here that was pulled out by the mass spec, and then the protein from which that came. So we've got this hypothetical protein here that was in both the ascending and the peak samples, and then these 2 here. What I should say now, actually, is that the protein levels didn't change between our wild types and our double knockouts here. So we're really looking at a differential loss of phosphorylation rather than degradation of the protein or something. So yeah, we had this hypothetical, absolutely nothing on that in TriTrip, of course. But we also had in the later samples, we had this kinesin family member C1 and this molecule here, which we were particularly interested in, and that's NOT5. So why did we care about NOT5? Well, NOT5 is what is known as a slender retainer. So TOR4 is another example of these. And slender retainers are super important molecules that hold slender forms and they're, as slenders. They act to prevent differentiation to stumpy cells. And it was Matthew Kayla, who heads up a lab in York now, who described NOT5 as a slender retainer. And what he found was when he depleted NOT5 in cells, he saw an accelerated production of stumpy forms. So this is parasitemia during an in vivo infection here. So when he depleted NOT5 in these cells, you could see accelerated growth, and this was in G1 during that infection. And you could also see this accelerated production of PAD1 positive stumpy cells. Cool, so how then do we bring together AKT-like, NOT5, and quorum sensing signaling regulation? So what we think might be happening is that under normal growth conditions, AKT-like is inactive, or indeed in the knockout, we haven't got AKT-like at all. So with AKT inactive, the slender retainer NOT5 is active. It's not phosphorylated at this point, and NOT5 holds these cells as slender forms. They're not going to differentiate to stumpy forms. Whereas upon exposure to the oligopeptide signal, AKT-like is activated. Don't know how, would love to. And with AKT-like now activated, it phosphorylates NOT5, this residue we found in the mass spec. This is an inactivating phosphorylation, and now with NOT5 removed, inactivated, differentiation from slender to stumpy forms can now proceed. So, to put that all together then, so we found these two kinases, AKT-like and AGCK, by GRiP-seq. We know that they're knocked down or they're knocked out, confers reduced sensitivity to oligopeptides in vitro, and it delays stumpy formation in vivo. We know that AKT likes catalytic residues, its lipid binding ability, in particular its T290 phosphosite, are important for the oligopeptide response. We've placed both the kinases upstream of all the previously identified quorum sensing components. And we think that how AKT-like, or one of the ways in which AKT-like might be working to regulate quorum sensing signaling is through an inactivating phosphorylation of the slender retainer NOT5. So, thank you very much for listening. This is everyone that has contributed in some form towards this project. So we've got everyone in Keith's amazing lab in Edinburgh. I'm very lucky to be able to work my best pals every day. We've got Mark and Lorena who help with the dipeptide stuff. My two incredibly talented undergrad students. They were so much better than I could have ever dreamed of being during my undergrad project. They were such a help. They were both amazing. Everyone in Nottingham who helped with the GRiP-seq stuff. Federico in Lancaster who started this off. Matthew, who's been a great help with answering my many questions, and particularly with the mass spec analysis, and then with the mass spec facility folk in Edinburgh. So yeah, thank you very much again, and I'll try answer any questions that you might have. Thank you. Questions?
Yeah, that's really nice.
Sorry.
So the way you draw it, you suggest the two kinases operate a single pathway. Do you know that?
No. That's just the way I've drawn that. I mean, we already know that the signaling pathway is certainly is non-linear. We know it's kind of branched. So I don't know exactly, is this one part? Is there two different branches that are kind of converging later? I don't know whether they're on that kind of same exact same pathway. It'd be cool to know if there were something happening, but don't know.
The peptides talked about NF, which came out of a
Yes.
screen of synthetic peptides, right?
Yes.
Has anyone ever looked at what the parasite proteases can generate? Do they generate NF if you expose them to serum?
So a PhD student in our lab, Mabel Tessie, she certainly started some kind of preliminary stuff with the mass spec stuff, trying to see what was getting generated. I think Keith had a look and hadn't found NF, of course. What I will say is, I mean, I'm definitely not standing here saying NF is the dipeptide. We were looking at very kind of small libraries. We were just kind of, things are important. It'd be cool if we could look at all of them. But yeah, I think what NF is really useful for is more being like a tool to kind of help probe this response. We're still using it at reasonably kind of high concentrations. It'd be cool if we could, get something that's maybe working at a lower concentration.
Can I ask a follow-up?
Go for it, yeah.
So the transporter in the parasite that brings those.
TPR89, yeah.
Yeah. What do you know about it? Is it possible that it's not actually, it's bringing peptides into the cell, but is it possible that it's actually detecting relevant peptides and sending a signal directly? So it's not the peptide intracellularly.
Yeah, it's something that's happening, yeah.
The transporter is acting as part of the signaling pathway.
Yeah. So, I mean, Federico, who was the one that kind of characterized GPR89, he did kind of demonstrate in vitro that, that GPR89 can actually like transport oligopeptides into a cell. So we know it's definitely got that capability. We don't know kind of what's happening after that, really. We don't know whether that could be, so I guess in other oligopeptide signaling kind of systems, there's, let's say membrane-bound kind of kinases that can kind of trigger things. We don't know, whether that's some kind of transceptor activity. Again, this is something, that we can kind of start probing now that we've got this NF dipeptide. We can start asking, okay, so what is actually happening between GPR89 and these oligopeptides when they're transported? GPR89, it is a little bit tricky in that we can't delete it or anything, so it's not the easiest one to play about within the cell. But certainly things we can, start to look at kind of selectivity. Is there a selectivity in the transport there? What's happening with oligopeptides? It's all the really cool questions, isn't it, that we don't know the answer to yet.
Hey.
Hi.
What role does cyclic AMP in the end actually play in this stumpy differentiation? So you can give this membrane permeable cyclic AMP and things happen. But where in the signaling pathway that we're now uncovering does it actually fit?
So, oh my goodness, we're going back to older papers here. So cyclic AMP, it peaks in the cells somewhere between slender to stumpy formation and then decreases as stumpies are formed. I think the original experiments that were done, they'd compared hydrolyzable and non-hydrolyzable forms of the cyclic AMP. So it was more the product. It wasn't the cyclic AMP itself that was triggering the slender stumpy differentiation.
That's how I remember it.
It was in hydrolysis product, AMP.
Yeah.
What happens then? I think is it AMPK has also kind of been implicated in slender to stumpy differentiation, which I think detects AMP. No, am I talking nonsense here?
I don't think that the cyclic AMP dependent kinase is involved. No.
Cool. That's as much as I think. I'm sorry.
Because there's still an unresolved thing. There is this peak of cyclic AMP, which suggests a role for it. But what generates that cyclic AMP?
Oh,
for instance. So there are cyclic AMP cyclases, generating it, but they need to be activated by something. So
no idea.
It's still very, very unclear.
So for your initial dipeptide screen, did you check all the 400 combinations of dipeptides?
Oh God, no. No, we are very, very kind of limited.
How many did you check?
So what we did was we went from Federico Rojas's original work where he'd screened a library. I can't remember how many he'd checked, but he had certainly seen that dipeptides that had an asparagine at the N-terminus seemed to be the best one. So we just tested asparagine followed by the rest of the combinations that we could get. So we're very, very limited. We kind of saw what it looked like. So NF was by far the best. It looked like asparagine followed by hydrophobic residue seemed to be the best. But it's very limited kind of screen that we were doing.
Estimate sort of the Z-score for the like how much was this one the best out of the ones you tested? Like, was there a
by far?
No effect, and then you had.
There was a lot of them that were no effect, or there were ones that there was an effect, but it wasn't. It looked like it was something else that wasn't slender to stumpy differentiation. We weren't seeing them arresting at the right phase or expressing at those proteins. So we saw a big difference between those, but we're only looking at 20. So
when you say these are dipeptides, do you literally mean that it's a dipeptide? It's just an asparagine. Okay, so it's very, very small. What was the concentration that you put on?
We were still reasonably high that we were working with. I think we were in the micromolar. We're still it went to 250 micromolar. Certainly, we're still kind of getting the effect. And we'd hoped on. We'd like to make something a bit more potent than that. But there's a huge number that you can screen.
we will briefly go online for, so there are a couple of questions. Frederick, if I've unmuted you, if you could ask your question.
Okay, so do you hear me?
Yes.
Okay, great. Okay. Hello, everyone. And thank you for your great talk, Christy. It was really fantastic. I have a question actually on the, I think it was Ari's comment about the MPK and so on. So you mentioned, if I got it right, that your mutants are not any more sensitive to cyclic AMP. So they don't differentiate into STUMPy when you add cyclic AMP. So my understanding is that they should be downstream of AMPK. So maybe I don't
If I don't know, if you knock, so AMPK is still one of the molecules that if you knock it down, I think the cells become less resistant to, no, so, wait a minute, less responsive to the cyclic AMP.
Because cyclic AMP should activate AMPK, because Ari mentioned that indeed they may be transformed into AMP into the cells and to activate AMPK. So if your mutant are insensitive to AMPK, it means that they are downstream of AMPK. AMPK could activate them, for instance. Maybe I don't get it right, but it's my feeling.
So I think I could be wrong. I think AMPK is one of the ones, if you knock it out, you don't see them responding as much to cyclic AMP.
Yeah.
One of the, actually, maybe I've got another slide here.
Because if they would be upstream, if they would be upstream of AMPK, if you add AMPK should be activated and the signal should go on.
Yeah, I don't know for the, well, this is the, well, perhaps this could be how things work with AMPK, because we know that AMPK is well, yeah, I'm trying to think. I'm sure when you're not doing AMPK, they're less responsive to, I don't know. How I thought it might be working.
The data are not clear yet, but presume what is assumed is that cyclic AMP activate AMPK, one of its derivative, probably AMP, activate AMPK, and then you go on. And AMPK may be one of the very first kinase of the signaling pathway. And since your mutants are insensitive to AMPK, they should be between insensitive to cyclic AMP, sorry, they should be between AMPK and the rest of the pathway, of the signaling pathway. It's my understanding, but maybe I am wrong.
No, maybe I'm completely wrong as well. Amazing. we can get back to it.
It's always complicated. This I prefer metabolism finally. But anyway, it's a fantastic work. Congratulations.
Thank you.
Typical signaling discussion.
Yeah, I need to sit down with a bit of paper and say, right, what does this do and this do? Right, okay.
There's another question online from Jay Ganesh. Do AKT-like and AGCK mRNA levels stay up in fully mature stumpy forms? Also, are they upstream of the ZC3H2?
H20, yeah. Okay, so the mRNA. I can tell you what I saw on the protein level from my mass spec. So from the protein, it looks like the protein level in stumpies of AKT-like goes up a bit. It looks like the protein level in stumpies of AGCK goes down a bit. I'm not sure about the mRNA, but hey, we've got the protein. So.
Any other questions on the floor? Any questions online? There are no more. Thanks again, Kirsty, for a wonderful talk.
Thank you.
We'll all meet back on March 13th for another GRiP-ing seminar.